Hey, my name's Owen McDougal. I'm a professor and chair in the Department of Chemistry and Biochemistry. And my lab looks at everything associated with food, dairy, natural products. We cover everything from toxicology, forensics, to food and dairy process. So along those lines, we do work across probably a dozen different projects, and the goal is mostly to align students with the projects that they're inspired by. So at this stage, I've got one postdoc, research technician, in the process of hiring another postdoc. We've got 3 BMO graduate students, PhD students, chemistry master's student, and about half a dozen undergraduate students. So we run a pretty decent sized lab. We've got 2 different lab spaces, 700 square foot in one, about 350 square foot in another. We look at everything from potatoes, potato chips, French fries, milk, cheese, whey protein. We look at a variety of different plants that grow naturally within the Boise area, and same genus of plant that grows in Georgia. So we've got plants that poison people that they mistakenly eat, and then we do analysis of like blood and breast milk samples and other types of things. We're also looking into therapeutic development based on bioactive components of milk, and also bioactive components of the Veratrum californicum, and moving on to Veratrum parviflorum at this stage for cancer chemotherapeutics from natural products. We study things like wine and barley. So we're doing a barley germination study now with pulse electric field. So the thing I like about the biomolecular sciences program is we use physics, chemistry, and biology in just about everything we do. We got a project with Tiana. Tiana is something that hits the opioid receptors in the brain. It's about as addictive as heroin. It's sold over the counter in a lot of places as a homeopathic nerve calming remedy. And this came to me from a former student who went on, he's in medical residency in Missouri, and they just kept seeing patients that were higher and higher doses of Tiana, and they couldn't get off it. So it's very difficult to get people that are heavily addicted to opioids off. So we're studying what's actually in the products that are being sold to people. Pong pong is called the suicide tree or suicide seed. This was sent to us from doctors at Emory University in Georgia in order to do analysis on a patient who tried to commit suicide by ingestion of the seed from the tree. And we have blood samples from that patient that we can study the analyte within their system over time and help the doctors figure out how to identify more quickly from a blood sample whether they've been poisoned from this particular compound. And then Jared, who's on this line, Jared Seal is working on blood, breast milk, and plant products from Veratrum parviflorum that poisoned a variety of doctors and their doctor spouses. So 3 couples all poisoned by mistakenly eating Veratrum parviflorum, thinking it was a mountain onion off the Appalachian Trail. So they were all hiking the trail, poisoned themselves, ended up in the emergency room, and took a variety of blood and breast milk samples in order to identify whether we could get the steroidal alkaloids identified within their system. So those are some of the projects we do. Lately, I've been working on a food and dairy innovation center. So we've got a new building on campus, the Micron Center for Materials Research. And I've been in a fundraising campaign to build out laboratory space to create new labs within that building to study food and dairy research. And that's together with Lisa Warner in chemistry, together with me, Julie Oxford in biological sciences, Joanne Leidy in engineering, and Jim Browning also in engineering. So within our team, the Food and Dairy Innovation Center looks to be a transdisciplinary research space for science, engineering, and technology, looking to drive innovation in the food industry. So within that, our goals are infrastructure. I want to build out new labs and a new building and have a new space to do work. So shown on the bottom left is a picture of the lab space and its current configuration. 8 a.m. meeting this morning to talk about what the infrastructure would look like. It should be done by fall 2023. So we're hoping to move in pretty quickly. And then sustainability, we do a lot of work with dairy, wine, beer, and within that, it's fostering collaborations that provide internship opportunities for students. So I've got a third-year biomolecular sciences PhD student, Joseph Collins, that will do an internship with AgriPure in Minnesota this summer. He'll get paid very well to do that. He's got years of funding from dairy work to support his graduate work. So we try to align folks with projects that are funded by industry so that they don't have to take on as much of the teaching obligations unless I want to. So Food and Dairy Innovation Center, basically our focus is down here on the bottom with food processing, dairy processing, biomedical research, and also computer science associated with advanced manufacturing, advanced artificial intelligence type work. So we use a lot of instrumentation in our work. So if we study French fries, it's using mass spectrometry, near infrared spectrometry, whether it's liquid or gas going into the mass spectrometer. Our methods are simple. We do basic extraction, solubilization, and chemical analysis of analytes. Within that, we get complex spectra. We can use that in order to do qualitative and quantitative analysis. And we work a lot with industry. So having food physics in Boise has allowed us to have access to pulse electric field systems and be a real partner for their company, which is anything associated with food and dairy. So it's pretty cool how that works. Pulse electric fields allows for electroporation. It facilitates extraction of analytes or components from cell membranes. It can be used in anything from making potatoes, potato chips, French fries, to beer, wine, and other things. So pulse electric field is becoming an integral part of what we do in partnership with food physics. We do a lot of work looking at acrylamide, which is a probable carcinogen, and California Proposition 65 made that a huge concern regulatory-wise for the food industry. So there's a avenue there for funding and student work and support for students. We use a triple quad mass spectrometer in the Biomolecular Research Center to do the acrylamide analysis, which is really cool. This is a new instrument within the last year, $400,000. So it allows us 1 part per billion analysis detection. Dairy research, we do a lot of mid and near infrared spectroscopy, a lot of chromatography, circular dichroism, differential scanning calorimetry, and we're looking at a variety of different aspects, protein structure, protein denaturation, solubilization, things of that nature. We're working with the computer science department to implement machine learning into analysis of spectra acquired from either mid or near infrared spectroscopy for things like dairy protein identification, quantification, and also acrylamide quantification. We're looking at partnerships with material science and engineering, looking at plastics. So renewable plastics, biodegradable plastics, food grade plastics, in collaboration with Scott Phillips. Biomedical applications, these are mostly with industry partners, looking at things like glycomacropeptide, lactoferrin, and a variety of others. So within the biomedical applications, we're doing everything from extraction, separation, characterization, and bioactivity analysis. So we have access to cell culture facilities and can do a variety of cell culture work. Within dairy, we're also looking to get more into microbiology. Food safety, food security, microbiology comes in and plays a heavy role there. We're looking at things like food toxicity and poisoning of individuals, steroidal alkaloids from plants. This is a picture of Madison Dirks, a first-year biomolecular sciences PhD student, I guess second now, up at Bogus Basin. So this is a local photo with local plant. Shown in the right is the Veratrum parviflorum from Georgia. Similar types of steroidal alkaloids out of those plants. Similar process, cut it up, dehydrate, extract out the components of interest, bioactivity, assessment and evaluation, separations, characterization. So we look at individual analytes, components, and we try to put together what a profile for a plant would look like. And we can study those analytes within blood and milk samples. So within that, getting identical and characterization of analytes is very important. We also look at beer and wine, and with pulse electric field, you can improve the extraction characteristics from the grapes in order to make better quality wines, clearer whites and redder reds. We use scanning electron microscopy to look at the yeasts that are used in the winemaking process, and this is in partnership with engineering. And we can also use pulse electric field to accelerate germination. In the bottom right are pictures that we just shared with Anheuser-Busch yesterday. Pulse electric field can save water during processing, reduce energy cost, and improve efficiency of processing. And we're looking at climate change issues as well. So the impact of smoke in rangeland and forest fire smoke on potato growth. So setting up collaboration with University of Idaho, the Parma Field Station, and then also the Food Technology Center in Caldwell, Idaho, and then building out labs on the third floor of the new building. This area for proposed Food and Dairy Innovation Center, we're looking to take over about half of the one side, the west side, north end of the third floor of the new building. And that's the lab that's available in person. And anyway, that's what we have going on in the lab these days. So thanks for your attention.